All right, let's see. Is this recording? Doesn't look like it. No, it is.
Congratulations. That's good. That's really good.
Okay.
And now I'm looking for the right one. Yeah. No?
All right, so let's get started. Today we're going to be going over fatty acid degradation. Here are your learning objectives, and we'll keep going forwards. And now this is what we've done already, and this is where we're going. And so, this is not working. All right, here we go. We're going to start off today with a case. We have a 46-year-old female who works at the ICU as a nurse. She is known to have type 2 diabetes. And after she pulls a double shift, she has trouble breathing and feels too sick to drive home. She stops over at the ER to visit a friend and say, you know, can you check me out, see what's happening? And so her vitals, her respiratory rate is rather increased, so it's about 40 breaths per minute. She has a low blood pressure, very hypotensive, so 99/52 millimeters of mercury. Her blood glucose is above the limit of detection for the finger stick analyzer. So she's diabetic. She's having some diabetic problems with her glucose regulation in her blood. So nothing too surprising so far. So we have some blood values. Her lactate is really high. Her pH is also very low. Her bicarbonate is very low. Her oxygen, partial pressure of the oxygen in her blood, also low. And her creatinine is very high. So we see that she has low blood pressure. Her body's in an acidosis situation, so she's very acidic. She has low bicarbonate. She has low oxygen. She has a low pH. She has a lot of lactate because without that oxygen, you can't go forward. And so you're stuck after glycolysis going into an anaerobic condition. And with a high creatinine, she's going into acute renal failure. So something happened over that double shift that has put her body really not in a good place. So it's lucky that she was actually at the hospital already. And we'll see what possibly could be going on with her, knowing that she already has this background of diabetes. So we'll start off talking about how we have our lipids, and then we transport them into the mitochondria to go through oxidation. So with lipids using as fuel, they are about 2.5 times more potent in producing ATP than glucose. If you fully oxidize a lipid versus glucose, you'll get a lot more of that ATP per gram than glucose. To oxidize fatty acids, you will generate more molecules of water, so your body will become actually more hydrated by going through lipid degradation. And fatty acids are oxidized in times of low blood sugar, fight or flight mechanism, hunger and starvation, fasting, that kind of thing. And also during times of growth and as a child developmentally, you also will rely on lipids as fuels for certain key points through development. So as we mentioned before, lipids are usually stored in our body as triglycerides. And so to use those triglycerides as fuel, we first have to mobilize them. Our adipose tissue is where we store the triglycerides, but we have to get those triglycerides to other body parts, to other organs, so that they can use that energy. So we use enzymes called lipases, which really means just breaking a lipid. It will sequentially hydrolyze triglyceride into glycerol and 3 fatty acids. So these enzymes will act at the oil-water interface within the lipid droplet of the adipocyte itself. So here, if we have the adipocyte, we have this lipid droplet, which in actuality takes up the majority of that cell, but for just purposes of visually, it's very small here in comparison. We have our triglyceride being broken down to a diglyceride and a monoglyceride and then to glycerol. So they'll have 3 sequential workings of lipases. And they will each generate these fatty acids, which will be transported out of that lipid droplet and will be exported out of the cell. The glycerol itself can also be used, and that goes straight to the liver for either glycolysis or gluconeogenesis, whichever the body really needs. The fatty acids themselves are exported to the blood and plasma where they're bound to albumin, and this helps transport them to all the tissues that need them. It can go straight to the liver, or it could go to any other tissue for oxidation. As I just said, glycerol is released to the blood and is taken up directly by the liver, where it serves as a gluconeogenic substrate. So hormones will trigger the release of these fatty acids from the triglycerides. Very similar as we've seen before, this is controlled by a hormone type receptor where it could either be glucagon, cortisol, ghrelin, testosterone, growth hormone, epinephrine, or norepinephrine. So as you can see, there's many different types of hormones that will act on these receptors. So these adipocytes contain multiple receptors for each of these different hormones, and they are active under different conditions. For example, glucagon and cortisol is helping to regulate that blood sugar. The ghrelin is for hunger and starvation. Testosterone, growth hormone, is really for growth and development. And epinephrine and norepinephrine is really for that fight or flight mechanism. Either way, they're all coming in and activating a G-coupled protein receptor on the cell surface of those adipocytes. And it leads to a signal transduction cascade where that protein kinase A will phosphorylate a lipase. Here it's triacylglycerol lipase. The name of the lipase isn't important. There's many of these inside of the adipocyte. The main point is that it activates those lipases to turn the triacylglycerol into the diacyl and then into the free fatty acids in the glycerol. And those go straight through out of the adipocyte into the plasma. Yes?
I just have a general question about this topic. So basically, depending on which hormone is sitting in the receptor,
Yes.
basically are all these processes happening, or is protein kinase only doing the triglycerol? Like, so for example, if glucagon bound to it, and then that process happens, but are all the other processes happening as well?
So all of the so when you say all the other processes, let me just double that. You mean all those other hormones or all of this cascade?
Like the cascade.
So the cascade will happen once that hormone activates that receptor. So no matter which hormone it is, they'll all act through a receptor and go through the entire cascade to release those fatty acids and that glycerol. Now, it doesn't matter which hormone does it. They will all work through the same mechanism. They all converge on this protein kinase A. The exact receptor and the G proteins that are assembled with it will depend on which hormone it is, but they all converge down to that same protein kinase A, leading to the breaking of the triglycerol into the free fatty acids and glycerol.
So like protein kinase A is doing a ton of phosphorylation on a ton of different things.
Right, protein kinase A is very non-specific. It will phosphorylate a lot of different substrates. We've seen that also when we were talking about glycogen. Protein kinase A, it converged on protein kinase A as well, the same signaling pathway. So as you can see, glucagon, which also will regulate glycogen, also will regulate fat homeostasis. So it will use the same hormone, work on the same protein kinase A. It's a protein kinase A that then finds its substrates, and that will lead to the rest of the signal. So once we have our lipases, they will then sequentially remove those 3 fatty acids. And as we talked about on Friday, these triglycerides are chiral molecules. There are specific lipases that will remove the first lipid from the first fatty acids from the third fatty acid from the second fatty acid. They will recognize the chirality of this triglyceride. I don't really care if you know which ones do which. Lipase is all that's really important. The only thing is that we're releasing those fatty acids. They go straight to the blood, they bound to the albumin, and they get transported. So we have, as I said, the fatty acids are bound to albumin. They'll go to many tissues. They can go to brain, they can go to liver, they can go to spleen, whatever. And once they're in the tissue, they'll go under fatty acid oxidation, where we're essentially going to be converting that long chain fatty acid into segmented units of acetyl-CoA that will go through the citric acid cycle and produce your carbon dioxide and your water, and of course the ATP, which we all want. So as you can see, this just converges into some of the pathways we've already talked about. However, glycerol only will go to the liver. There it can serve as 2 different substrates. It can either be shunted into glycolysis and produce pyruvate, and then from pyruvate you just keep going, as we talked about, or you can use glycerol to produce glucose through gluconeogenesis. So glycerol itself is acted upon by something called glycerol kinase, which phosphorylates it. You produce L-glycerol 3-phosphate, and then this will eventually turn into some of the intermediates that we're familiar with glycolysis, our dihydroxyacetone phosphate and our D-glyceraldehyde 3-phosphate. These are, again, the 2 end products that you get at the end of that investment phase of glycolysis. So we just will skip all of those steps by going straight from glycerol to there. And again, this is only happening in our liver. So once we have our free fatty acids separated from the glycerol, glycerol will do its thing. The fatty acids will go throughout the body, whereas glycerol, it's only going to the liver. Those free fatty acids will go through a process called oxidation. And this starts in the cytoplasm, however, will finish in the mitochondria, specifically in the mitochondrial matrix. The final product of this entire process is acetyl-CoA, which we can put right directly into that citric acid cycle and then transfer our electrons through the electron transport chain and get our ATP out at the end. So with the start of the oxidation, we have our free fatty acid. It's in our cytoplasm. We need to first activate it so that we can actually go through the actual process of oxidation. And so we do this through an enzyme called acyl-CoA synthetase. And so we're essentially going to be using this ATP-dependent acylation of coenzyme A. So we're essentially just attaching that fatty acid onto coenzyme A. Now, in doing so, you can see here's our ATP, where our adenosine is there, and then we have our triphosphate. And then we essentially will take that fatty acid and attach it onto that adenosine and a phosphate. And then from there, we will take that acyl group, that fatty acid, and put it directly onto our coenzyme A. To do this entire process, we are breaking up that ATP that we initially started with. However, on paper, it looks like, oh, we're only breaking up this one ATP. In actuality, because of the, we're actually attacking and removing that 2-phosphate there, and we're leaving only an adenosine, a phosphate, and a fatty acid, it's really like the equivalent of a loss of 2 ATPs. The amount of energy that is required to do this reaction is as if you use 2 ATPs. So once we have the acyl-CoA, we have our fatty acid on coenzyme A, we're then going to let it get into that mitochondria because all of those beta oxidation, all those oxidation enzymes are located in the mitochondrial matrix. So we attach that acyl group, that fatty acid group, onto a molecule called carnitine, and this will transport it into the mitochondria as an acyl carnitine. And so this is done through a transport protein, which will take carnitine with this acyl group on it. As it moves that in, another carnitine gets bumped right back out. And so we have this cycle so that we can continually bring in the acyl group, that fatty acid group, and then remove the carnitine from the mitochondria to go back outside to pick up another fatty acid group. Once inside, that acyl group is going to be put right back onto that coenzyme A by another acyl transferase. So we're just moving this fatty acid acyl group around and around in the mitochondria from the cytoplasm to the mitochondria because all those enzymes are in that matrix.
There's a big difference between 1 and 2?
No.
1 is 1 and 1.
One is located in cytoplasmic, 2 is located in the mitochondria. It's just that simple. They have some sequence similarity, but really it's that location that really makes the difference. So once we're in the mitochondria, we have options for degradation. The most predominant and most common type of oxidation of fatty acids is what's called beta oxidation. And so beta oxidation is a cycle of 4 different reactions that just gets repeated and repeated until you chew up the entire length of that fatty acid. So it's a cyclical removal of 2 carbons to produce acetyl-CoA. So if we start off with a fatty acid, we will then go through 4 reactions, remove an acetyl group, put it on CoA, so we get acetyl-CoA, and then we have a fatty acid side chain that is 2 carbons shorter than what we began with. This goes right back to the beginning of beta oxidation, and we go over and over and over again until we chew up that entire length of that fatty acid. Yes?
So do we have to memorize all those steps?
We will talk about all of those steps, yes. And those, this is the only oxidation pathway that you need to know any of the enzymes for. The other oxidation pathways, just the conditions, and we'll talk about that as we get to the other pathways. So for fatty acid beta oxidation, there is a cycle of 4 different types of reactions. We start off with a dehydrogenation, so we're removing a water group essentially. We're going to then hydrate it, so add water back in. Then we're going to dehydrate it again, and then we're going to cause a thiolytic cleavage. So we're going to be using a sulfur to cleave something. And so we'll talk about all these steps as we go along. We're going to yield an acetyl-CoA, an FADH2, and we're going to make that fatty acid CoA shorter by 2 carbons. And so as you see, as the cycle goes over and over and over again, all of these acetyl-CoAs, they'll add up, and all these FADH2s will also add up as well. So the first reaction is the acyl-CoA dehydrogenase. And so this will catalyze the removal of 2 hydrogen atoms from the alpha and beta carbon. So remember that alpha carbon is the first carbon after the carbonyl, and the beta is the second one. And so we're going to end up with a trans alpha beta unsaturated acyl-CoA product. So we have this double bond here between that alpha and that beta carbon, and it is in the trans location. So, excuse me. The enzyme that does this acyl-CoA dehydrogenase, contains a FAD molecule itself as a cofactor. And then the hydrogen of the beta carbon will be transferred directly to that FAD, and have an enzyme will be bound to the FADH2, which we then can, of course, transfer those electrons to the electron transport chain. So these bound FADH2s, which are bound in that dehydrogenase enzyme, will directly transfer those electrons to coenzyme Q in the electron transport chain. So you skip all the steps before. You skip complex 2 because primarily that FADH2 is bound directly to that dehydrogenase enzyme. We can't transfer the electrons to complex 2. It goes directly to coenzyme Q to ubiquinone instead in the electron transport chain. So once we have that, that first reaction, we move on to the second reaction. This is a hydration reaction by an enzyme called anoyl-CoA hydratase, where we're going to be adding in a water, and we're going to be breaking that double bond that we just formed, but we now have transferred on an OH group, which was not there before. So the whole purpose of forming that double bond was to eventually add a hydroxyl group onto that beta carbon. Part 3 now, we have that acyl-CoA dehydrogenase comes in again. It will remove the protons, we create some water, and we will also transfer electrons to NADH. And now you can see we have a new carbonyl bond. Yes?
I assume the answer is yes, but should we know the names of all of these, like L3, hydroxyl, CL6, all those?
The 4 enzymes in beta oxidation, all the other enzymes we're going to be talking about today with oxidation, no.
I mean enzyme, I mean the name of.
Oh, the names of like the intermediates? No. You don't need to know those names, no. Just know basically what kinds of reactions, like the dehydrogenase is a dehydrogenase reaction. We're taking away water and as in the form of hydroxyl group, where as form of hydrogens, we're moving those electrons. The hydratase is adding water, that kind of thing. So these electrons are being transferred to NADH. So what do we think we're going to do with them? We're going to put them in the electron transport chain. So now at the very end, we have this 3-keto acyl-CoA. So we have these 2 carbonyl groups, one of which is on that beta carbon. And so now we're going to use an enzyme called thiolase to form thiolytic cleavage. So this is the last reaction of beta oxidation, and it is a nucleophilic attack by the sulfur on coenzyme A to the electron poor keto carbon of the 3-ketoacyl-CoA. So we will cause a release of an acetyl-CoA, and then we will have this new acyl-CoA being formed. So this enzyme is called thiolase. It's a unique reaction, and it will come up in some other pathways as well. So it's an important enzyme to remember. So here we see that we start off with this 3-ketoacyl-CoA. So we have these 2 ketone groups right there, where this is the original carbonyl carbon, and this is our new one on the beta carbon. We have a new CoA coming in, and this CoA will attack this second carbonyl group. We will remove off those first 2 carbons and produce acetyl-CoA. And then we have this new acyl-CoA, which is 2 carbons shorter than that first one. The reason why this is called beta oxidation is because we're cleaving between the alpha and beta bond. So the new carbonyl carbon right there, that was the old beta carbon. And so that is why we're calling it beta oxidation. So looking at the energy yield from beta oxidation, we're going to start with a fatty acid called palmitate. This is a 16 carbon saturated fatty acid. So there's no double bonds. We can go through beta oxidation. So to go through and full oxidation of this molecule, we'll go through the beta oxidation pathway 7 times. So the initial activation of the palmitate is the loss of about 2 ATPs. You will produce the 8 acetyl-CoAs when you cut this off into 2 carbon CoA bits, and feeding those acetyl-CoAs into the citric acid cycle and then down the electron transport chain, you're going to yield about 80 ATPs from those. With the FADH2s that we produce from the first step, we're going to produce about 10.5 ATPs. From the oxidation that occurs in the third step, we'll produce about 7 NADHs. You'll get 17.5. The grand total for a palmitate, to produce palmitate to go into carbon dioxide and water for ATP is 106 ATPs. So way more ATPs you're getting out of this oxidation of one molecule compared to what you'd get out of glucose. So let's have a little review question. Which of the following is true about beta oxidation of fatty acids? Has everyone had a chance to answer? Okay, so majority has oxidation occurs in the mitochondrial matrix. A couple of the other answers chosen were the first two, catalyzed by lipases, or oxidation requires ATP. So the correct answer is oxidation occurs in the mitochondrial matrix. The actual process of beta oxidation is in the matrix. The initial activation, that is all occurring in the cytoplasm, and that is the step, the activation before it even gets in the mitochondria, that will use ATP. None of the enzymes that are actually involved in the process of beta oxidation, none of them will use ATP at all. And then this can occur in almost every cell, not just the liver, and so that's that question. So before we move on to the other types of oxidation and the conditions in which they are used, are there any questions about beta oxidation? Okay, so we're now going to go through a series of different other types of oxidation reactions. They are not the main ones, and they're only used under certain conditions. However, because of the wide variety of lipids that we have in our diet and that we make in our body, they're all necessary and they're all very important. So with these mechanisms, really what I'm more concerned is you can associate the name with the type of lipids that it degrades and that kind of thing, and where they occur. You do not need to know any of the enzymes involved in any of these oxidation mechanisms after beta oxidation. So one type of fatty acid that's very common are the unsaturated fatty acids. These are the ones with the double bonds. So first we'll look at the simplest case, and this is the monosaturated fatty acids. So they only have one double bond. So with this kind of, when you encounter this kind of fatty acid, we can go through beta oxidation starting from the very end where there's the CoA, where the coenzyme A is attached to, until you get to that double bond. All of the enzymes involved in beta oxidation cannot recognize a double bond and use it as a substrate, so we have to go through this different pathway. Now this occurs in the mitochondria, so all the enzymes involved in beta oxidation are already there. The fatty acid that has the double bond doesn't have to be transported anywhere. It can stay put in the mitochondria. Instead, it will be going under its own little scenic route back to becoming a saturated fatty acid. So we have this double bond, which we will then use an enzyme called cis-delta 3-anoyl-CoA. Again, not important, you know the name. And then this will essentially turn the cis double bond into a trans double bond, which is very similar to one of those intermediates that is involved in beta oxidation. And then you'll just continue on with normal beta oxidation as if this was just the natural intermediate. So it's very easy to do a monosaturated fatty acid. It's just a little sidestep from your basic beta oxidation. However, when we do a polyunsaturated fatty acid, this requires a lot more work because there's multiple double bonds. You're essentially going between different types of oxidation pathways. You're starting off with beta oxidation until you get to that first double bond, and then you're going to be using those isomerases and some other reductases to constantly move double bonds and move carbons, move hydrogens, so that you can end up with intermediates that can be fed into the beta oxidation pathway. The oxidation of polyunsaturated fatty acids is also occurring in the mitochondrial matrix, so all the enzymes required for both this pathway and for the beta oxidation pathway are all in the same area, and it makes things just move a lot smoother. So another option we've seen our double bond containing fatty acids. Not all of our fatty acids are going to be even numbers. So we have looked at a couple slides. We all are ending with even numbers. When we looked at the palmitate, 16 carbons long, you could divide that into acetyl-CoA, 2 carbon units. You'll go through fatty acids. You'll go through making acetyl-CoA in even number intervals very easily. However, with odd number carbons fatty acid, you can't end up with acetyl-CoA at the very end. And so you have to have a different pathway being involved. So with an odd number fatty acid chain, if you were to go all the way to the end, eventually you'll get acetyl-CoA at the end, and you'll end up with one propionyl CoA. So here we have our very end of our beta oxidation of an odd chain fatty acid. We have the CoA, we would do our thiolase, our thiolated cleavage, and then we end up with acetyl-CoA, and we end up with propionyl CoA. Propionyl CoA has 3 carbons. So we're not going to cleave off 2 carbons because then we would just have a lone methane group, and we don't want that. So we're going to keep this propionyl CoA and then convert it into something called succinyl CoA. And so propionyl CoA carboxylase will do exactly that. You start off with propionyl CoA, you go through a series of steps, you end up with succinyl CoA. And we've seen succinyl CoA before. Succinyl CoA is just a succinate added to that CoA. We can put that into citric acid cycle. We can get energy out of this. The enzyme that will do all of this requires a vitamin B12 as a cofactor. And so this, of course, if you're deficient in vitamin B12, you're going to have problems with this type of reaction. Yes?
So normally you would have end up with 2 acetyl-CoA, and now we convert 1 over.
Right, so majority of the fatty acids that we consume are even numbered. However, there are ones that are important for our diet that are odd numbered. And so when you get to that, when you do beta oxidation in an even one, you'd end up with the last reaction. You have 4 carbons, you split into 2, and you end up with 2 acetyl-CoAs. With an odd number, you can't do that. And so you end up with 3 carbon unit, and then you just turn it into the succinyl CoA instead. This requires vitamin B12, which is a coenzyme called B12 cobalamin, which is very unique. It is one of only 2 molecules in our body that actually uses the element cobalt. And so cobalt is a trace element required by us primarily for this reason. And the actual production of this very large molecule used as this coenzyme is done by the gut bacterial symbiotes. You can get some of it from meat that's ingested and from fermented food like kimchi as well. And so there are multiple routes of getting this in. And so without the cobalt, you won't be able to do those odd number fatty acids. So we've looked at fatty acids that are fully saturated. That's our beta oxidation. We've looked at our odd numbers fatty acids, and we've looked at our unsaturated fatty acids. The last one we'll look at is branched fatty acids. So branched meaning you have your chain of your carbons, and then you have all these side carbons coming off of them. So this is called a branched fatty acid. And this undergoes a pathway called alpha oxidation. So in beta oxidation, we're cleaving at that beta carbon. Alpha oxidation, we're going to be cleaving at the alpha carbon. This is the only fatty acid oxidation pathway that does not occur in the mitochondria. It occurs in a organelle called the peroxisome. And this pathway, these fatty acids cannot physically go through beta oxidation. The enzymes will not work on them. You won't be able to actually produce any intermediates that actually will result in the production of acetyl-CoA. And so it is also a step-by-step process. We're not going to go through all the enzymes. However, eventually the goal is to remove acetyl-CoA chunks and other pieces of that fatty acid until you reach a section of the fatty acid that can eventually go through beta oxidation, and then it'd be transported back into the mitochondria and will work there. So we'll have a quick review question. So with this type of fatty acid, which type of pathway should be used? right, it looks like everyone stopped responding. We have majority at the PUFA oxidation, and then something that's not really showing up what values it is, but equivalent to, what is that, 12% saying monosaturated and beta oxidation. So the correct answer is you need both beta and PUFA oxidation. You're going to use that beta until you get to that first double bond. Until then, that's just simple beta oxidation. Once you get to that first double bond, then you see that these have three, this would be an omega-3 fatty acid, so there's three of those double bonds right in a row at the very end of that fatty acid. You're then going to take over and use that PUFA oxidation. And so alpha, we're not going to use that one because it's not branched, and then it's not monosaturated, so of course we wouldn't use that pathway either. So with oxidation pathways, it all depends on what that starting fatty acid is, where it's going to be transported to, what enzymes are going to work on it, and then the goal at the very end is, of course, get those acetyl-CoAs, push them through the citric acid cycle.
So the one of PUFA is trying to convert from the cis to trans?
Yeah.
From trans to cis?
No, cis to trans. So the majority of our, like we talked about on Friday, the majority of our dietary fatty acids are all going to be in cis. However, in the workings of beta oxidation, we do produce a transient trans orientation to help in the chemistry, the organic chemistry involved in actually going through the oxidation. And so those PUFA oxidation pathways and those mono oxidation pathways, monounsaturated fatty acid oxidation, those will also make and convert those cis into trans double bonds so that you can then go with forward.
So will it go through beta to get double bonds and then through PUFA, whatever, and then back to beta?
Right, it'll eventually go back to beta once you get those trans orientations back. With something where it's like this, where there's those three right in a row, it's going to go back and forth, back and forth, back and forth until you get to the very end of the molecule. All right, so any other questions before we move on to the last topic for today?
With alpha oxidation, how is it not? So we're trying to get it to be not branched. How is it not branched at the end with the racemic acids? But still it's branched at the end.
So we're taking off pieces of acetyl-CoA. So, and then the CoA will keep going. Eventually, we're going to have sections where there's regions where we can do beta, like a chunk like that, until you get to the next branch. With something like an alpha oxidation, it takes a little more time because the alpha and the beta oxidation pathways are in two separate organelles. We have the peroxisome doing the alpha oxidation and the mitochondria doing the beta oxidation. So as you get to a region where it can undergo through beta oxidation, that can't occur in the peroxisome. It has to be transported out of that peroxisome, back into the mitochondria, remove it, but then it gets stuck. It can't do those branch. So then it goes right back over to the peroxisome, remove the sections until you can get to an area where you can do beta oxidation, and it goes right back. So this alpha oxidation is a little bit slower than if you were just talking about like the PUFA oxidation or just a standard straight beta oxidation, just because of all the movement of molecules between cellular locations. Okay, so the last topic for today are ketone bodies. And this is something that we're going to relate back to our case at the very beginning. So further tests with our patient revealed that she had high levels of glucose and ketones in her urine, as well as very high levels of ketones in her blood. Normal range for ketones in blood is about 0.6 millimoles per liter. Hers is way past that at 8 millimoles, greater than that. It's also noted that she is severely dehydrated and that she is going to be starting on IV saline. Now, high levels of ketones and high levels of blood sugar usually do indicate someone is dehydrated because of a proper osmotic balance with sugar and ketones in the blood, which typically shouldn't be at those high levels. And so as more water is drawn out into the urine, because of the high levels of glucose and ketones in the urine as well, the patient is going to get more and more dehydrated. So when our bodies undergo fasting, whether it is intentional or during a starvation type situation, our carbohydrate intake is too low. The oxaloacetate levels in the citric acid cycle fall so that the flux through the cycle becomes impaired. And this causes levels of acetyl-CoA levels to rise. And so under these conditions, when you have multiple copies of this acetyl-CoA around, these can then be combined in something called acetoacetyl-CoA. And this is essentially like the reversal of a thiolase reaction. So remember that thiolase reaction, you're cleaving off 2 carbon chunks, and whereas here we're putting them right back together. So here we have this reversal of the thiolase reaction. So that acetoacetyl-CoA can go through a series of reactions, which we're not going to talk about all of the chemistry and the enzymes involved in which, but it will produce a lot of intermediates until it gets to here at the very end, our acetoacetate and our hydroxybutyrate and acetone. These final 3 are called ketone bodies, and these are synthesized in the liver when we have the starvation mode and these high levels of acetyl-CoA are made. This acetoacetyl-CoA is made throughout the body. However, this function and all these enzymes are occurring in the liver, so they have to be transported to the liver, and they're converted into these ketone bodies. Acetoacetate and D3-hydroxybutyrate can interconvert between each other relatively easily. However, acetoacetate can only fall into acetone. Acetone can't go backwards in this reaction. So these 3 ketone bodies, the whole function of this is trying to remove those acetyl-CoAs because they've built up, and we can use some of these for energy as well. Because remember, during these conditions, our citric acid cycle is essentially paused. It can't go any further because we've depleted our intermediates. And so the beta-hydroxybutyrate and acetoacetate themselves can be used as fuel. They go through and transfer they will transfer from succinyl-CoA into succinate, and will help to replenish the TCA cycle and produce acetoacetyl-CoA, which can be acted upon by our friend thiolase and produce more acetyl-CoA. And so the idea is that when times become better and we can now replenish our citric acid cycle, we then can use this acetyl-CoA to then produce energy again. Acetone itself cannot be used for fuel. That is something that just has to be eliminated, usually through urine. So during, like I mentioned, some of this is only going to be happening in our liver cell. However, it's also linked very much to our fatty acids. So when we have our fatty acids being released from our adipocytes, they'll go into the blood and into our liver cells. We will then produce it from our fatty acids, acetyl-CoA, and under the conditions of starvation and fasting and that kind of thing, we're producing our ketone bodies. Our ketone bodies can be pumped back into the blood, and this is why we can actually physically measure it in our blood draws. And then into the blood, it can go to cells to try and get them back into the citric acid cycle when we have the conditions that are right and we have those TCA cycles replenished again. We can use that for energy. And so they will be transported to some of the most important organs that we need to keep functioning: our heart, our renal cortex, and our brain cells. So the whole idea is we're under these poor energy conditions. We're trying to get some substrates to those really important organs so that when we have our TCA cycle replenished and active again, we can then really go forward and we can use them as energy.
So we have those 3 ketones that go into the mitochondria for acetyl-CoA, go into citric cycle.
Only 2 of them will. The acetone will not go in. It's only the acetoacetate and the beta-hydroxybutyrate. The other one, we can't use that for energy.
And where do they enter in the system?
Right. So they are going to be helping to replenish our succinate, and then we're also producing more acetyl-CoA. It's a little bit counterintuitive because the whole formed because we had too much acetyl-CoA, but then they produce acetyl-CoA again. So it's really producing this acetyl-CoA so that once it's replenished, we can actually go through again. The idea is that we're not having too much of a net loss and that we can get back what we initially invested. So when conditions are right, everything's going to go forward. Everyone will get energy again. Everyone will be happy. Ketone bodies also will form during diabetes. So when we have the changes in diabetics such that glucose is not being either taken up into cells or glucose is too high, and it will dampen the effects on the regulatory effects on adipose tissue such that fatty acids are more released and that they will go into the liver and ketone bodies will be formed. The glucose also is not properly working and regulating. There's no proper regulation on that liver either because of the high levels of glucose, and it will cause the citric acid cycle to slow down. This forms those ketone bodies. Now, if this happens over a prolonged period of time, these ketone bodies can build up to levels that are very dangerous, which can lead to a drop in blood pH and eventually to coma and death. So the formation of these ketone bodies is actually pretty quick. If you have a slowed down citric acid cycle due to fasting or dysregulation by glucose during diabetes, within a matter of an hour or so, you are producing a significant amount of ketone bodies. If this goes over a longer period of time, that's when you're going into those ketoacidosis. So bringing it back to our patient, being diabetic, she needs to be very aware not to skip meals and to stay hydrated. And during her double shift as a nurse, she was very busy and didn't take any breaks and didn't drink, and she became dehydrated. This potentiated she was essentially fasting in a little bit of a starvation mode. She's diabetic to begin with. So this caused ketones to build up in her system and would eventually lead to acute renal failure. So the good part is it's pretty much reversible. With aggressive fluids, insulin, bicarbonate, and norepinephrine, she made a full recovery within 2 days. So of course, there's many physiological changes that happens with diabetes. This is just one of them. So a little review question. Which ketone body cannot be used for fuel?
CD died this time.
What?
Every example you ever give, like every patient died.
Oh, no, the first one with the baby didn't die. I wasn't going to do that to you guys. I don't think our last patient died either. It was only the mitochondria guy. The last one, no. I don't think he died. We'll say he didn't. All right, so about 96% of the class say it's acetone. Glycerol itself is not a ketone body. That is released during triglyceride breakdown. It's a component of triglycerides, not a ketone body. The other two, acetoacetate and beta-hydroxybutyrate, those can be used as fuel, and it's only the acetone that cannot. So to summarize, triglyceride degradation includes the lipolysis of the free fatty acids from glycerol, both of which are a fuel source. And then we have multiple pathways for lipid degradation, and it all depends what you use and what that fatty acid is, whether it's saturated, unsaturated, branched, straight line, whatever. The ketone bodies will form under stress conditions and can be used as fuel. However, too much can lead to ketoacidosis and lead to some severe consequences. So any questions with today? All right, so next time we're going to do the exact opposite. We spent today breaking down those fatty acids. Next time we're going to build up the fatty acids and talk about why we actually build them when we can just eat them. So that'll be next time.
Quiz on Wednesday, right?
Yes.
Can the answers to the study guide questions for 32 and 33 be posted?
Yes, they will be.
Okay.
Yeah.